Welcome everyone to this UCL Lunch Hour lecture on From Gene Deserts to Druggable Pathways in Inflammatory Bowel Disease. My name is Dr. Charlie Murray. I'm a consultant gastroenterologist at the Royal Free Hospital and UCLH, and also chair of gastroenterology at Cleveland Clinic London. It is my honour and pleasure to introduce today's speaker and my friend James Lee. Dr. Lee is a clinical sciences group leader at the Francis Crick Institute and an honorary consultant gastroenterologist at the Royal Free Hospital. He studied medicine at Oxford and completed his PhD at the University of Cambridge. In 2014, he was awarded a Wellcome Intermediate Clinical Fellowship and spent half of this award at Harvard University before returning to Cambridge in 2018 to establish his research group. He joined the Crick in 2021, and his work has been recognised by numerous prizes, including the Julia Bodmer Award from the European Federation of Immunogenetics, the Sir Francis Avery-Jones Medal of the British Society of Gastroenterology, and the United European Gastroenterology Rising Star Award, and in addition to that, a Lister Prize. Before we begin today's lecture, I just want you to know that we will have some time at the end of the lecture for questions, and these can be submitted at any point during the talk by going to Slido, which you should have in a link. And if you don't, you can search that up, Slido, and enter the event code #discovery. With great pleasure now that I hand over to my friend James for his talk.
Thank you, Charlie. It's a great pleasure to join you this afternoon. Thanks very much for the invitation. I think it's great to do these sort of public engagement talks, and hopefully we will keep this at a level that everyone can sort of follow along. So, as well as introducing me, I also just want to introduce the real sort of heroes of this piece. So I'm going to be talking to you today about some work that we've published over the last few years, and in particular, one piece of work that was published just before the summer, and that was largely driven by these two individuals who are in my lab. So my lab is based at the Francis Crick Institute, and I'm fortunate enough to work here, but also, have a great working relationship with all of the gastroenterologists and the team up at the Institute for Liver and Digestive Health. So this team is what has actually facilitated a lot of the work that I'm going to talk about. And actually, I'm in a really unique position in terms of being a clinician scientist with both, access both to patients and also access to the sort of research facilities at the Crick that enable us to do the science we like to do. So what is inflammatory bowel disease? You heard it mentioned a couple of times when Charlie was given the introduction. When we talk about IBD or inflammatory bowel disease, what we really mean are two separate diseases. One is Crohn's disease and one is ulcerative colitis. And collectively, these diseases now affect about 1 in 100 people in the UK. Both of these are incurable, immune-mediated diseases. So when I explain this to my patients in clinic, I explain that essentially your immune system has got its wires crossed. So instead of fighting off coughs and colds and keeping you well and healthy, it's actually attacking the bowel by mistake. And that attack leads to damage and inflammation and ulceration within the bowel. And this is a really serious condition. So, left untreated, this can drive complications that ultimately can require urgent and sometimes life-saving surgery. Importantly, we're not talking about irritable bowel syndrome. A lot of people get IBD and IBS confused for obvious reasons. They're obviously, not only do they give you similar symptoms, but the names are very similar. But if you do a colonoscopy on someone with irritable bowel syndrome, this is what you will see. Nice, healthy mucosa where you can see a nice lacy pattern of blood vessels, and nice pale background mucosa. Unfortunately, this is not what you'd see if you did a scope on someone with inflammatory bowel disease. I won't leave this too much up for too long because I know people are having their lunch, but you get this inflammation and ulceration that actually causes physical damage to the gut. So who gets IBD? Well, unlike many of the diseases, that we treat in modern medicine, this is not a disease that you get when you get old. This is a disease that typically occurs in young people. So the peak incidence occurs somewhere between the ages of about 16 and 40 years old. And I think it's worth just pausing for a moment and just think back to the sort of things you were doing, when you were between 16 and 40. Maybe some are still in that window. Chances are you'll be thinking about the time that you finished your education, or maybe when you settled down, and got married, or maybe even started to have a family, or, or got your first job. And IBD can be hugely disruptive to all of these really key life events. But the thing is, IBD has been described as a global ticking time bomb, and none of what I've shown you so far is the reason for that. In fact, the reason for that is this. So this is the global prevalence of IBD. And we've known that this has been going up for a long time. So for about the last 40, 50 years or so, we've been aware of a rising incidence in Western Europe and North America. What we're now starting to see in the last 5 or 10 years is an explosion in IBD in parts of the world where we never saw it before. So Sub-Saharan Africa, the Middle East, and the Far East, for example. And collectively, what this means is we are just seeing this progressive, steady, and sort of inexorable rise in the prevalence of these diseases. So to put that into context for you, when I started my PhD in 2008, it was estimated that maybe 1 in 200 people in the UK were affected by IBD. Recent estimates put that number at about 1 in 100. So actually, the incident or the prevalence of IBD has doubled even within the last 15 or 16 years. And that creates a major problem. It not just in terms of, the need for healthcare resources, but actually also the fact that we don't have a single treatment that works for everybody. So this is data from a whole series of phase 3 studies that have been done in ulcerative colitis, one of the forms of IBD. And on the left-hand side of the dotted line are the early remission rates in patients who got the drug. And on the right-hand side are the early remission rates in patients who just got placebo. And what I'm putting this up to illustrate is that while we have lots of different therapies, they're only inducing early remission in somewhere between about 15 and 40% of patients. So the majority of our patients are not getting better quickly on any of the drugs that we have to offer. So what that effectively means is we need better drugs. But there's a problem there too, because if you take the drugs that are going into clinical development, only about 1 in 10 ever actually become approved therapies. So in other words, 90% of them fail at some stage in the development process. And the vast majority of these fail at a stage called phase 2. And phase 2 is the point at which you figure out the dose that you need to give people to make them better. So what this means, it's not that the drug doesn't do what it's intended in terms of blocking a kinase or mopping up a cytokine. What it means is doing that in someone with Crohn's disease actually doesn't make them any better. And I think what all of this tells us, between this hugely high attrition rate in drug development and also the fact that our current therapies only work in 15 to 40% of people, is that fundamentally we still don't understand these diseases well enough. Otherwise, we wouldn't, to put this very simply, be targeting the wrong pathways when we develop new candidate drugs. So the question then is, how on earth can we understand IBD better to start improving some of these numbers and ultimately to end up with better therapies that we can offer our patients when we see them in clinic? Well, the approach we've taken to this is actually to use disease genetics. And there's a couple of important reasons for that. The first is that over the last 20 years, genetic discovery in IBD has been incredibly successful. So when I started in science, we knew of maybe 4 or 5 regions in our DNA that were directly involved in causing these sorts of diseases. We now know of over 400 regions in our DNA. So that's been an incredibly successful enterprise, but it's not just simply a stamp collecting exercise. Because if you take a drug that's going into phase 1 study, and if that drug is targeting a mechanism that's implicated by disease genetics, that drug has a far higher chance of actually working. So originally it was estimated to be about 2-fold higher. In fact, if you really understand the pathway and your drug hits that pathway, that's probably about 6-fold higher. So you could take your 10% success rate and turn it into something much more like a 60% success rate, if only we knew exactly what the right pathways were to target. But there's a really big problem. And the problem is that most of these genetic associations, which we found through these large genome-wide association studies, do not lie in the coding sequence of genes. So in other words, they're not having predictable effects on protein structure or function. In fact, the vast majority of them seem to lie close to or within these elements called enhancers. Now, enhancers are a bit like the volume dials in your genome. They're able to turn up or turn down the amount of a nearby gene that's being made. And don't forget that every cell in our body has the same DNA. So what makes a muscle cell and a liver cell are the different genes that are turned on in those respective cells. And all of that is controlled by things like enhancers. So these are really critical control elements. But if you want to study them, you've first of all got to figure out, well, what cell type does the enhancer work in? And secondly, you've got to figure out, well, what the gene is that the enhancer is regulating. And only when you've got those bits of information can you start to think about what effect the genetics might be having on this process that ultimately contributes to someone developing a disease like Crohn's disease. So what that means is that at the moment, we understand where all the genetic risk lies, but we have very little idea about how it is actually leading to the development of disease. And so while there's an amazing potential for genetics to transform both our understanding of why people get diseases like IBD and also potentially how to treat it, at the moment, that potential is largely unrealized. So where does that leave us? Well, actually, it leaves us with very long lists of genetic associations which we know are directly involved in these diseases. So the thing is, we can pinpoint where the important stuff is happening, but we don't really understand what's going wrong. And people have slightly unkindly said, well, that's about as useful as just sitting down and reading the telephone directory, because it doesn't tell you anything about the disease process. And when you look in this in a bit more detail, you can kind of understand that position. So here are some associations. We know the chromosome they're on. We know the site on the chromosome. We even know some of the genetic variants that may be contributing. But at that point, our certainty ends. And at that point, we're kind of left guessing as to what might be going on. So let me show you this. Here's an association where the authors of this paper, and I was one of those authors, we guessed at what the two key genes might be that may be driving this disease association. But look, there are 63 other genes at that site. So how confident are you that we got this right without really understanding anything about what the genetics is actually doing? Perhaps even harder, here are some associations where in fact there are no genes. So these used to be called gene deserts. They're large stretches of our DNA where there are no genes obviously there. So how do people handle this when we find these? Well, to be honest with you, the way people have typically handled it so far is they've ignored them, because we've got enough genetic associations where we can identify a candidate gene and come up with a story about what might be going on, that when we find things where there's not obviously anything going on, we kind of just ignore it and move on. So the approach my lab take at the Francis Crick Institute is not to ignore this, but actually to treat this as what it is, which is effectively a treasure map. Because what this is telling us is where all the important stuff is in the genome. It tells us exactly where it is. The only thing we don't know is what the treasure is. We don't know what the mechanism is by which this is actually driving disease. And so my lab at the Crick and prior to that in Cambridge are specifically interested in going to actually try and figure this out using the genetics as a starting point. So with that, I want to talk to you about a paper that we published a couple of months ago. It is, in fact, one of these gene deserts. It's one of these regions where there would have been a 0 next to it in terms of how many genes there are in the candidate region. But the really interesting thing is that this region doesn't only increase your risk of getting both forms of inflammatory bowel disease, it also increases your risk of getting a whole load of other inflammatory diseases as well, including ankylosing spondylitis, which is an arthritis that principally affects your spine, and also primary sclerosing cholangitis, which is, an inflammatory liver disease that can lead to liver failure and in fact increases your risk of both liver and bowel cancer. So finding something like this tells us a couple of things. The first thing it tells us is that this region must be fundamentally important, because why else would a same set of genetic variants in this gene desert increase your risk of 4 or 5 different diseases? The second thing it tells us, since these are all immune-mediated diseases, but they obviously affect different tissues, is that if this contains one of those enhancers, it's probably an enhancer that's doing something in an immune cell. So that was our starting point. And in fact, this is that association. So each of these dots here is a genetic variant, and the higher they are up the y-axis, it tells you the more they are associated with the disease. So you've got this peak of association here. This is with inflammatory bowel disease. But underneath this peak, there aren't any genes. So we figured this must contain an enhancer, and the enhancer was probably an immune cell enhancer. So actually, that's what we went looking for. And in fact, you can find active enhancers using a method called K27 acetylation ChIP-seq. Now, the details don't actually matter, except to say it enables us to find enhancers that are switched on. And what we were struck by when we looked in at exactly the site of this association is that this seemed to be functioning as a very strong enhancer in one type of white blood cell, so-called monocytes, which are really important, by the way, for driving a lot of the inflammation that we see in diseases like IBD. But actually, we couldn't find any evidence of this region being an enhancer in any of the other sorts of immune cells that you would typically associate with the development of these diseases. The other thing is that, although this seems to sit a long way away from any nearby genes, the way our DNA is organized in the nucleus of every cell is not like this. It's not a long straight line. If you peeked in with a magnifying glass, it's much more like a bowl of spaghetti, where in fact everything is jumbled up and there are things from one side that may be touching things on the other side. And actually, we can do experiments to figure out what are the contact points. And when we did that, surprisingly, it wasn't the nearest genes, which a lot of people had thought it must be, but actually it was a gene that in genomic terms was really quite a long way away, called ETS2. And we could show that this enhancer physically touched the promoter of this gene ETS2. The other thing we could find by going and looking in healthy donors who either carry 2 copies of the risk allele or only 1 or perhaps none, was that the more copies of the risk allele you had, the higher the level of this gene ETS2 was in your monocytes. So it seems that having more of this gene in your monocytes, which can be genetically determined, seems to play a direct role in the development of multiple diseases. But of course, the next obvious question is, well, what on earth does this gene ETS2 do in these monocytes? And sometimes doing biology is actually very simple. So, if you have a fuse in your house and you want to know which, does this fuse, control your TV or your lights or your oven, the easiest thing sometimes to do is just to turn it off and actually then go and see whether the telly has stopped working or the lights have gone off. And actually, we can do a similar thing in biology. So using CRISPR, we are able to take primary human monocytes. So these are cells that have come out of a healthy donor. We can put in the machinery to delete that ETS2 gene, and then we can culture them under conditions that turn them into an inflammatory macrophage. And the question then is, well, what effect would that have by turning off this one gene? And strikingly, we found that by turning off just a single gene, we were able to turn off a whole series of different inflammatory functions in macrophages. The macrophages lacking ETS2 were not able to become activated. They lost the ability to phagocytose. They lost the ability to migrate. They lost the ability to make pro-inflammatory cytokines. They even lost the ability to make reactive oxygen species, which is the thing that fundamentally damages the mucosa. When I'm doing an endoscopy and I see ulcers, this is the thing that drives that. So you've got multiple pathways, all of which seem to directly be involved in the disease process, and all of which require the ETS2 gene in order for these processes to occur. So in the same way that sometimes to figure out what something does, you can turn it off, the other thing you can do is actually turn it on in a setting when it wouldn't otherwise be on. And this is actually really quite important from a biological perspective, because lots of genes are essential for different processes. If you imagine a signaling pathway where the first gene signals to the second gene, which signals to the third gene, and so on, if you take out that second gene, the signaling pathway can't function. But just having a bit more of that second gene won't make the pathway function more if there's the normal amounts of all the other downstream stuff. The only situation in biology where having a bit more of it tends to be sufficient to turn the pathway on is when you have something that's a sort of central regulator or a master controller of the pathway. So we set about to do that experiment. We can this time take, again, primary monocytes, but this time we're going to turn them into non-inflammatory resting macrophages. And then we can make RNA that actually encodes the ETS2 gene. This is done using exactly the same technology by which your COVID vaccine is made. But instead of making spike protein for COVID, we're making the ETS2 gene. And then we can put that ETS2 gene into these macrophages, or a control that's the same length and the same nucleotide composition, just to control for that process. And then we can begin an incredibly low grade inflammatory response just to see what happens. Do cells loaded up with ETS2 behave any differently? And what we've decided to do was go back and look at those pathways that we knew ETS2 was essential for. And strikingly, what we found was that every single one of those pathways turns on when you turn up ETS2. And the really exciting thing about this, and this is one of the beauties of this method, is we can control how much RNA we put in. So we can either put in a low dose or we can put in a higher dose. The low dose here is in gray and the higher dose is in black. And you can see that every black circle is to the right of the gray circles. So what that means is not only is this sufficient to turn on inflammation, but it can do it in a dose-dependent manner. So the more ETS2 you have, the more inflammation turns on. So that then actually meets the criteria for being a master regulator of this process, because it is not only essential, but it is entirely sufficient to switch on this whole gamut of different inflammatory functions in macrophages. And in fact, it became even more surprising, because if you take a signature, a gene signature of inflammatory macrophages, this is from single cell RNA sequencing data, from the inflamed macrophages within the gut of patients with Crohn's disease, the vast majority of those genes turn on just by turning up ETS2. And in fact, that includes all of the genes that we currently target our best therapies against. So just to put that in a different way, you can take a resting macrophage and turn on a single gene, and the cells then look like they've just come out of the gut of someone with Crohn's disease, which is really quite remarkable. But of course, when we are doing these sorts of studies, as I said to you before, the genetics isn't stamp collecting. The aim of the genetics is to then identify genes, and that in turn enables us to then identify the pathways. And the pathways are really where the money is at, in terms of actually being able to really understand the disease better. And actually in IBD, this has been a very successful enterprise, because we have previously shown that a process called autophagy is directly involved in the development of Crohn's disease. And that solely came out of genetic studies where half a dozen autophagy genes were identified as being involved. The reason that's relevant is that is the benchmark, half a dozen autophagy genes within a pathway. So what we said, first of all, was maybe if this ETS2 pathway is as important as we think it might be, maybe some of the other genetic hits are actually also pointing at components of this pathway. And so we actually went back to our experiment where we had turned off ETS2, because we've now got a nice long list of all the genes that seem to be directly under the control of ETS2. And we just simply looked in that list of genes that go down when you turn it off and said, are any of these known from genetics? And remarkably, loads of them were. So, we're well beyond the half a dozen autophagy hits now, because you've got over 20 IBD genes that are robustly linked to IBD that are directly under control of ETS2 and within this pathway. In fact, you can go further than that, because you can start to think about doing pathway analysis. And again, the details of this don't specifically matter, except to say what it enables us to do is look at the relative enrichment of any given pathway we like, compared to, within the genetics of IBD. And it enables us to do that comparatively. So we can say, is autophagy more enriched, for example, than T-cell signaling? So we decided to do that experiment. And actually what we found was that the ETS2 pathway, however we defined it, was probably more enriched for the genetics of IBD than pretty much anything that's ever been described before, including all the success stories like autophagy and TNF signaling, but also things like T-cell activation and IL-10 signaling. So then the question then comes is, well, might this provide us with a therapeutic opportunity? And of course, in terms of thinking about a therapeutic opportunity, the key question is this pathway actually switched on in the diseased tissue? And actually, we were able to go and actually test that using publicly available data. So this is single cell RNA sequencing data. Each of these tiny dots is a cell, and these are all the myeloid cells, so the monocytes, the macrophages, and the dendritic cells. And here is a mixture of some that have come from Crohn's patients and others that have come from healthy individuals. And in fact, if we separate out the ones that came from the healthy controls from the ones that came from Crohn's, it's very obvious that the sort of the myeloid landscape in Crohn's looks very different to what you see in health. And the two things I really want to call out are this blue cluster of cells here, which are cluster 1, and this green cluster of cells here, which are cluster 2. And these just don't exist in healthy controls. The reason that's relevant is when we then looked at the genes that are regulated by ETS2 and we added up their expression in each cell, what we realized was these two clusters were the cells that seem to have ETS2 signaling very strongly switched on. And in fact, when we used a whole series of different methods to identify what these two clusters were, we quickly realized that these are the inflammatory macrophages and inflammatory monocytes that have been reported to be hugely expanded in IBD and produce many of the cytokines that we target our best therapies against. But as much as I'm a gastroenterologist and I want to look after patients with IBD, at some point we also had to remember that IBD wasn't the only diseases that were associated with this gene desert. So what about some of the others? So actually, we were able to go and find, again, using publicly available data, that if you take the tissues, the affected tissues from these different diseases, whether that's the synovium from the joints of patients with ankylosing spondylitis or the liver tissue from patients with PSC, you could see the hallmarks of ETS2 signaling, the footprint, if you like, being very strongly switched on in these tissues compared to healthy controls. But we wanted to actually go a bit further into this. And again, we were very fortunate because within the ILDH at UCL, we also have great collaborators on the liver side. And actually, they also have access to explanted livers, so livers that have been taken out when patients came for a transplant from diseases like PSC. And what we decided to do was an experiment where we could actually combine the sort of single cell sequencing data with the spatial data that we would get from a histology slide, for example. And this is called spatial transcriptomics. And what we did was compare some PSC samples with some healthy control samples. And now we could go and identify all the cell types. Don't worry too much about reading all of this. Suffice to say that we could identify the sorts of cells we expected to see. Now, PSC is a disease that specifically targets the cholangiocytes, the bile duct cells. And so what we were able to do was take every single cholangiocyte and then essentially just draw a circle around it. And that circle could be different sizes. And what we found was that in health, as you may go to bigger and bigger sizes of circles, you get increasing numbers of non-inflammatory macrophages and inflammatory macrophages, as you might expect. But in PSC, there seems to be this huge enrichment of the inflammatory macrophages compared to the non-inflammatory macrophages. In fact, you can do this analysis in a slightly different way. You can take every single bile duct cell, every single cholangiocyte, and you can draw a line to the nearest macrophage, whether that's inflammatory or non-inflammatory. And again, it didn't, for the non-inflammatory macrophages, they were very similar in terms of their distance from the cholangiocytes. But when you looked at the inflammatory macrophages, they're much, much closer to those cholangiocytes in PSC than in health. So not only do you have this big expansion of these inflammatory macrophages, but they are clustering around the bile ducts. They're very close to the target cells. And then finally, what you can do is take every cholangiocyte and then draw almost like a sort of target where you drew ever increasing rings around that cell. And for each ring, you can actually count up the number of ETS2 regulated genes as a sort of surrogate for how much ETS2 was switched on. And what we found was that this score was highest for those macrophages that were closest to the cells. So this is the sort of, it's the metaphorical equivalent of being, caught in the act as you're burgling a house with the swag bag on. You're at the site, you're doing the things you shouldn't be doing, and you're clearly directly contributing to the trouble that's going on. So The thing is, we've known for some time that macrophage inflammation is really important in all of these sorts of inflammatory diseases. And we know that for lots of reasons. We know that because macrophages produce the cytokines against which we target our best therapies. We also know that because they also make all of the chemicals that start to damage the tissue and ultimately lead to that sort of picture I showed you at the beginning of all the inflammation. Now, while we recognize that targeting single cytokines maybe only works in up to 40% of people, one thing we're starting to realize is that if you put two drugs together, so you don't just target TNF, but you target IL-23 as well, then actually that is better than targeting either alone. And that's because inflammation isn't driven by single molecules. In fact, often there's more than one thing happening. And, soon enough, we will realize that if two is better than one, then maybe three will be better than two, and maybe four will be better than three. And what all of that means is at some point, we're going to get to the realization that what we really want to do is not, give a patient 20 different monoclonal antibodies, but actually find a target that sits somewhere at the top of this pyramid that may actually enable us to turn the whole thing down. And that's what we think we found, because the ETS2 is undoubtedly the gene that is the master regulator of this process in inflammatory macrophages. So does this provide us with a therapeutic opportunity? Well, it turns out that there's no sort of off switch within the ETS2 protein itself. We'd hoped there would be, but there wasn't. And so we decided to take a slightly different approach. So there's an amazing data set that exists in the US where they have taken half a dozen different cell lines, and for each cell line have treated that cell line with one of 6,000 different small molecules. And what they've then done is deposited the genes that change in response to that small molecule. So you've got a compendium of over 30,000 signatures of over 6,000 different drugs. And what we did was we took that list and we said, do any of these look like they may be acting by switching off ETS2? Because we now know exactly what that looks like from a transcriptional point of view. And actually, to our great delight, several of them did. And interestingly, when you top these up and group them according to their mechanism of action, the one that sits at the top are MEK inhibitors. Now, that's really interesting because MEK inhibitors are actually already licensed for use in people. They're used for cancer, they're used for neurofibromatosis. They have not been used for inflammatory diseases before. The other thing that was reassuring was all of the inhibitors in the red boxes here are genes that are known to be upstream of ETS family members in terms of a signaling cascade. The other thing that was very nice was actually we also pulled out several of the therapies that we currently actually use for patients with IBD. So we decided to do a really simple experiment. We made our inflammatory macrophages again, but this time we didn't crisp edit them. We just put on a MEK inhibitor to see what effect that would have. And sure enough, it completely copied the effect of turning down ETS2. So all of those pathways that we know are ETS2 dependent go down when you treat with a MEK inhibitor. But of course, treating macrophages in a dish is a pretty artificial experiment. So we wanted to try and get a bit closer to what the sort of inflammation is that patients have. And one of the things that you can do is you can actually take biopsies from patients with active inflammatory bowel disease before they get any treatment. And those biopsies can be cultured for up to 24 hours in the lab. And in that time, they will continue to release all of the pro-inflammatory chemicals and cytokines that they were releasing when they were part of the patient. And again, the great strength of the setup that we have, and through my links with Royal Free and through ILDH in the Division of Medicine, was that we could actually get biopsies from patients at the Royal Free, and we could bring them down to the Crick in order to do this experiment. The nice thing about this experiment is you can put drugs into the media in order to see what effect that drug would have on any of the processes. So the first thing we did was measure a whole series of inflammatory cytokines, and we compared the effect of putting in a MEK inhibitor with the effect of just putting in a vehicle control. And what you can see is you see significantly less of all of these inflammatory cytokines being released when you treat with a MEK inhibitor, including all of the current best targets for IBD treatments. The other thing you can do is at the end of this time, you can actually look at the genes that are being expressed in these biopsies. And what we used was a biomarker that's been well validated to give a nice readout for how much inflammation there is in a biopsy. And we could show that the amount of inflammation was also significantly reduced by treating with a MEK inhibitor. Now, we've had lots of patients asking us for these sorts of treatments. One thing that we should say is that MEK inhibitors are great, but they can't be taken for long periods of time because they have on-target side effects, because MEK does things in other tissues. So in fact, one of the things we're now working on in the lab is to try and make these much more targeted and therefore safer as a way of delivering them just to those inflammatory macrophages that are actually contributing to all the problems you get with IBD. So this was the paper we published. It's open access if anyone wants to read it. It was published just before the summer. You may have actually heard about this already. This created a huge amount of media interest, and in fact has continued to create a huge amount of media interest internationally, because actually we think for the first time we found one of the most central pathways that goes wrong in these sorts of diseases, and we may have found a way in which we could intervene therapeutically to try and correct it. So I hope I've convinced you that actually using genetics is a very productive and useful way of trying to learn new things about diseases. It can not only tell us about those molecular mechanisms, and I didn't even talk about this side of the work we've done in terms of understanding why the genetic variant disrupt and increase the activity of that enhancer, but it also enables us to understand what goes wrong transcriptionally, and ultimately to understand how this affects the behavior of immune cells in a way that leads people to have a much higher chance of developing these diseases. And of course, all of that is done with one eye on the idea of maybe if we really understand these better, we may be able to find better ways of actually treating these sorts of diseases in the future. So with that, I'm going to stop. I just want to acknowledge some of the key players again. So Christina and Christoph in my lab are amazing scientists who have driven much of this work. Leah's a great collaborator from Berlin who helped out a lot over the last year or so. I've got an amazing lab at the Crick, and actually I'm supported not only by people at the Crick, but actually people across UCL from within the Division of Medicine, and actually also at ILDH, and actually also from the NHS side as well. There were really helpful collaborators, including my chair today, who helped us obtain those biopsy samples so that we could test out our hypotheses. Of course, science is a team enterprise, and we couldn't have done this without huge number of collaborators across the UK and Europe and North America. And in fact, that spanned not only academia, but actually also even into industry as well. So these are the people who funded the work. I'm going to stop speaking now, and I'm very happy to take any questions.
Thank you, James. Superb talk as usual. And great to hear it spoken so clearly. So we've got a few questions already. So I might kick off with one just because while the science is fresh in our brains, genetic variant we talk about, does this genetic variant have any benefit to humans? Because you told us all the bad stuff. And if not, why do we have it? Why would we have such a thing?
So that's a great question. And in fact, it's something that we really we take on in the paper to try and understand this. Now, most genetic variants that increase risk of disease are usually less than about 10,000 years old. And in fact, we had a great collaboration with the Ancient Genomics Group at the Crick, who also have affiliations to UCL, to try and understand when did this genetic variant actually arise in humans, and and why is it so common? Because unusually, this risk-associated variant is not the minor allele, it's not the rarer allele. In fact, it's the common allele. Many of us will carry this. And what we realized is that unlike the majority that arose within the last 10,000 years or so, this variant appears to have arisen at least 500,000 years ago, if not 1,000,000 years ago, and can even be found in Neanderthals. And what that means is there's been plenty of time for this to be selected out of human populations, but clearly it hasn't been. And actually, we went on to show that one of the things that this genetic variant does is, as well as just increasing ETS2 in resting monocytes, it particularly increases it in early responses to bacteria. And we're talking about within the first couple of hours of seeing a bacteria. So this is almost certainly an evolutionarily conserved way in which we could fire up our macrophages very quickly when you were inoculated with a bacteria because you'd fallen and cut yourself. And that may have made the difference between ultimately failing to control that inoculation, developing an infection, and before antibiotics dying of sepsis, or actually being able to survive. And we think that this is almost certainly why it's so prevalent. it probably also tells us quite helpfully that when we target this therapeutically, we probably want to turn it down. We almost certainly don't want to turn it off. And that's really important in terms of then thinking about how we target our therapies.
Yeah, I think that there's another question associated with that, actually. So if you were to turn this off, what downsides would we see?
So I think from our perspective, we don't want to. If you turned it off altogether, you might imagine that patients would be susceptible to bacterial infection, particularly given what we found about these sort of links to bacterial responses. One of the fortunate things, actually, as it transpired about finding that a MEK inhibitor was a useful way of targeting this, is if you want to turn something down, it's quite hard to do that if you have a direct inhibitor of the thing itself. So if we found an ETS2 inhibitor, finding a way to turn it down but not off would be quite tricky, because either you inhibit it or you don't. So actually targeting a regulator does enable us to potentially turn it down but not off. And so actually what we're now working on in the lab is finding a way where we can safely get enough of the drug into those macrophages to turn down this process, turn off the inflammation that's linked to IBD, but not leave those macrophages so susceptible, for example, to an infection.
And just a question about the MEK inhibitors as well. Is there any data available about responses of IBD in patients that have been on MEK inhibitors for other reasons that happen to have had IBD, and what has happened?
Yeah, so that's a great question. We spent a long time looking for that sort of data and we never found it.
Yeah.
I think one of the things is that MEK inhibitors aren't used widely in cancer. They're usually used as sort of adjuvant type treatments in cancer. And so while there will be patients who have been treated with them, there's just not enough that we know of who've had IBD in a way that would have then made sense. I think the other challenge, of course, is with MEK inhibitors, because they can't be used for long periods, you're usually only looking at a short snapshot. And of course, because IBD is an incurable disease, we would really want to be using something long term. And that, again, is why we're working to try and make this safer now.
Great. So we've got a general question, but it really ties into this, and I think we'll breed some more questions. So why do you think IBD rates have increased so much in recent years?
Again, that's a really good question. We know it's not the genetics. We know that the genetics has stayed stationary over the period of time at which we have seen these increases. So it must be environmental. I mean, the traditional teaching of IBD, which is almost certainly correct, is that you need an environmental trigger in someone who has genetic risk. So the way I explain it to my patients sometimes, it's a bit like firing a gun. So your genetics loads the bullets in the gun, and then the environmental trigger pulls the trigger. So if you have a gun that has no bullets in it, it doesn't matter how much you pull the trigger. And that's presumably why not everybody has IBD, because we're all exposed to a Western lifestyle. On the other hand, if you have a gun that's got lots of bullets in it, but nobody pulls the trigger, that gun also doesn't fire. So if you don't see the environmental trigger, you almost certainly won't go on to develop IBD. I think what this tells us is that the environmental trigger has to be something to do with Westernization, but that may be diet, it may be smoking, it may be emulsifiers, it may be pollution, it could be stress, early antibiotic use. There's so many different things it could be. And there's other research going on to try and understand what those triggers are. For us, actually, it's kind of easier actually to come at this from the genetics, because you can do the sort of mechanistic studies that are much harder to do with environmental studies.
Okay, and another question leading on from that. So you mentioned lots of things there, but you didn't actually mention food. So to what extent do you think food we eat has an impact on our guts and an impact on IBD?
So we know food has a massive effect on our microbiome, and we know that actually those bugs and viruses in our gut are the principal target of what's going on in IBD. It's the immune cells that are actually attacking those things, and the gut's getting damaged as a sort of bystander process. So diet does play a really important role. People have now started to look at whether, ultra processed foods contribute, or whether it's the amount of emulsifiers in food contributes. There's great research on this being done elsewhere in London. For example, Professor Kevin Whelan over at Barts is doing really important research in this area. As I say, we also sometimes actually use dietary therapy to treat IBD. So in Crohn's disease, you can use diet as a form of treatment. But interestingly, that works in Crohn's disease, but it doesn't work in ulcerative colitis. And we don't understand why that is. So as much as that we know the diet plays a really important role, we don't yet have a good enough mechanistic understanding of exactly what that role is in a way that could enable us to actually intervene therapeutically.
Do you think there'll ever be a time when diet and food will be able to activate or at least acts on these pathways to downregulate inflammation?
So that's a great question, and it's a great question because stay tuned, and that's probably the next thing that we're going to be writing up in the near future. So interestingly, it's very difficult to study diet and and the environmental trigger and the genetics together, but there turns out to be one genetic association with IBD that is actually a gene that is both genetically implicated but also is a sensor for diet. And in fact, we've done work on this, and we think this may be acting through the same pathway. So there may in the future be a way in which, using diet, we may be able to manipulate this pathway, probably not completely, but certainly in a way that may help in patients who have relatively mild symptoms. So that's ongoing work in the lab at the moment.
And really important, because of course that's what we would all aim for, wouldn't we, is to put ourselves out of business with dietary intervention. Now, this is a great question, a very bold one for you. Could the discovery of this gene that you published on now and have given us such a good talk on be used to eradicate or cure the disease?
That's a really, really important question. The ultimate ambition for everybody, if anyone who works in IBD and you say to them, like, in one sentence, tell us your goal, your ultimate ambition, the goal has to be cure. That is the fundamental thing that we're all aiming for. I mean, just to sort of wind back from that, though, we're currently in a situation where the best drugs we have only work in 20 to 40% of people. So cure is a long way away. But one of the things that you absolutely have to have in order to even contemplate the idea of a cure is actually an understanding of what goes wrong to drive the disease. So that's exactly what we're working for. Now, at the moment, the most immediate next application of this will be treatment rather than cure. But the more we start to uncover these pathways, and this won't be the only one, the more we can actually start to think about, is there a way that we could reset this actually back to the point at which, we did have tolerance to those bugs that when otherwise our immune system is now attacking and thereby causing damage to the gut.
Okay. And do you think there's a point in life in general? You talked about the spike in the prevalence of IBD between 16 and 40. Do you think there's something important about the pathway or merely the environment that means that's the age in which people get this disease, these diseases?
I think, that's a fantastic question. We really don't know. I suspect it's something to do with the environmental exposure to the environmental trigger at that sort of age. We know that there's a genetic component to that as well. So, we do occasionally see very young children who have rare, very damaging mutations, usually in single genes, that can develop IBD, within a few years of life. And so what we typically see is that depending on the amount of genetic contribution, it correlates with how early you develop your disease. Interestingly, there is a second peak of patients who develop IBD who develop it in their 60s and 70s. And again, that's not well understood either. But I think it's going to be a combination between the genetic trigger being you're most exposed to it, whatever it is at that age, and there's having a sufficient genetic burden that actually that's the point at which the whole process gets set off.
Okay. I'm going to be a little unfair because I know the answers are very difficult to this, but in your clinical practice, therefore, you talk about the host of environmental triggers that we've already mentioned today. Are there any, for example, for worried parents who both have inflammatory bowel disease, they have a child, they want to know how they can maximize their chance of their child not developing IBD, despite we would think anyway, that they have the gun loaded, they're waiting for the trigger to go. What would you say to them?
I think it's difficult. The first thing I always say to patients in this setting is that you don't need to be alarmed, because although if you have Crohn's disease, the chance of any of your first degree relatives getting Crohn's or ulcerative colitis in their lifetime is about 10%. So there's a 90% chance they won't. If both parents have it, it goes up to about 30%. But still, there's actually a greater chance they won't develop it. So in terms of things that people should do, we would, the triggers that we know are smoking. We know there is some literature around, mode of delivery, about whether it's better to have a vaginal delivery than a cesarean delivery. There's some literature around early life exposure to antibiotics. But actually, in all of these situations, you have to think about both sides of the equation, because if you're needing antibiotics as a child, it's usually because you're unwell. And I certainly would not be advocating not to give antibiotics in case it were to in some way modify your future risk. So I think all of these things have to be taken all together. I tend to reassure parents, because they usually are quite anxious, that this is not going to, be something that will automatically be passed on through everyone in the family, but also just to be vigilant. The one thing we do know is that, smoking, for example, is a very strong risk factor for Crohn's disease. So we would absolutely encourage people not to smoke if they have strong family history.
And how about ulcerative colitis, James? What do you say about that and smoking?
Yeah, so we know ulcerative colitis is perhaps the flip, that ulcerative colitis can be protective. In ulcerative colitis, we often see the disease arising in people who smoked and then stopped. So actually, that seems to be a bigger trigger than never smoking. So actually, I would advocate not smoking in the first place as the best way of preventing, IBD, although there is conflicting data from both Crohn's and ulcerative colitis. And again, this kind of underpins the fact we don't understand these diseases well enough, because we still have no idea why smoking might predispose you to one disease, but perhaps stopping smoking predisposes you to the other disease.
Okay, James, that's excellent. Now, I think we're going to wind up fairly soon. So I just thought if there was sort of the main take-home points from what was an amazing talk today, James, how would you summarize to our audience what they think they should take home from this?
Yeah, I mean, I think hopefully this is the sort of message we've tried to give to patients as well, is that I think this is a message of hope, because actually, the genetics has been incredibly successful, but for a long time, we've had associations that we just don't understand. This particular association that I've talked to you about today was actually first associated with all of those diseases over 10 years ago. So that's been sitting there in the literature for 10 years, waiting for someone to actually do these sort of experiments to actually figure out what's going on. I think one of the great things is that we've built up a toolkit in my lab over the years that now enables us to do that process much faster than would ever have been possible before. And actually, we're working really hard, and I've got a great team of people in the lab who are working really hard every day to actually try and develop better treatments, try and develop a better understanding of these diseases, in the hope that actually in the future, patients will have much better treatment options and maybe even a cure in a way that they currently don't have that.
Thank you, James. And everyone, you heard it here first, that Dr. Lee is going to cure Crohn's and colitis. We look forward to that. Listen, everyone, I'd like to thank you for attending this UCL lunchtime lecture. It's been a real pleasure for me to share this with my friend, Dr. James Lee. We look forward to seeing you at other lunchtime lectures. And for now, I will leave you with the rest of your day. Thank you very much.
Thank you.